For decades, scientists kept finding the same disturbing pattern whenever they compared human DNA across the world. The farther a population's ancestors had traveled from Africa, the more of humanity's original genetic variation seemed to disappear. Europe had lost part of it. East Asia had lost part of it. The first settlers of the Americas had passed through such severe population bottlenecks that their descendants carried only a fraction of the variation found in many African populations. But when researchers turned towards southern Africa, the pattern changed. In the genomes of several Khoe-San communities, they found branches of the human family tree that separated from other living populations so deep in the past that some estimates reached back toward the earliest stages of Homo sapiens itself. The discovery was quickly turned into a headline that sounded simple. Scientists had found the world's oldest race, but that headline was wrong. No living population is older than another. Every person alive today has ancestors who survived the same amount of time. DNA does not place humanity on a ladder, and scientists did not perform one giant study that ranked every race on Earth by genetic age. What the research actually revealed was more complicated and more important. Some living populations preserve unusually high genetic diversity and lineages connected to early splits in our shared history. Among the deepest of those branches are lineages found today in Khoe-San populations of southern Africa. That result did not create a hierarchy. It shattered one. For much of modern history, museums, textbooks, and popular documentaries place Europe near the center of the human story. African communities were often pushed into the background, as though the continent mattered only because humans had once left it. Genomics reversed that view. Much of it remained inside Africa, among populations whose histories had been repeatedly overlooked, divided, displaced, or studied without receiving equal control over the science built from their DNA. To understand what scientists found, we have to begin with a dangerous phrase, old DNA. When researchers say that a lineage is deeply diverged, they are not saying that the people carrying it are primitive, unchanged, or closer to apes. They are describing where a branch separates in a genetic family tree. Imagine two rivers that begin from the same ancient source and then flow in different directions. Neither remains frozen at the point where they separated. In the same way, a deep population split means that the ancestors of two groups became partly separated long ago, not that one group stopped evolving while the other moved forward. Genetic diversity is also not a simple clock. It can be influenced by population size, migration, isolation, admixture, natural selection, and sudden bottlenecks. A large population that remains connected across a wide region can preserve many genetic variants. A small group that breaks away may carry only a sample of those variants. If that group stays small, survives a disaster, or founds a new population, more variation can be lost by chance. This is why scientists do not calculate a population's age by counting mutations and announcing that one race is older than another. They reconstruct demographic history from many patterns spread across the genome. The global pattern begins in Africa because Homo sapiens evolved there. Fossils, archaeology, and genetics all place the deepest chapters of our species on the continent. Although the evidence no longer supports a simple story in which one tiny population appeared at one exact location and replaced everyone else. Modern humans probably emerged through long interaction among structured populations living across different parts of Africa. These groups separated, reconnected, exchanged genes, and developed traits over hundreds of thousands of years. Humanity may have formed less like a single tree growing from one trunk and more like several streams repeatedly dividing and joining. Most ancestry outside Africa descends largely from populations involved in major expansions roughly 50 to 70,000 years ago, though earlier movements also occurred. The groups that successfully spread into Eurasia represented only part of Africa's total population. They carried only part of its genetic variation. As their descendants moved farther, new groups repeatedly separated from older ones. Each separation could act like taking a handful of colored stones from a large jar. The new jar contains many colors, but not every color from the original. Take another handful from the second jar, and more colors may disappear. Geneticists call this broad pattern a serial founder effect. The effect can still be measured. Genetic diversity generally declines with increasing migratory distance from Africa. This does not mean people farther away are less healthy, less human, or less capable. It records the sizes and roots of founding populations. Europeans and East Asians carry less within-population genetic diversity than many African groups because their ancestors passed through ancient bottlenecks outside Africa. Indigenous peoples of the Americas experienced additional bottlenecks as small populations moved through Northeast Asia and eventually entered the Americas. Isolation and later disease, warfare, and colonial violence changed some communities even further. Yet the map is never perfectly smooth. The Middle East has been a crossroads for movement between Africa, Asia, and Europe. South Asia contains enormous diversity created by ancient settlement mixture and long periods of endogamy. Oceanians carry Denisovan ancestry at levels not found in most other populations. Europeans and Asians carry Neanderthal ancestry inherited from encounters after some modern humans left Africa. Africans themselves are not one genetic unit. The continent holds more population structure and deeper differences than the simplified categories used in many global surveys. That last point is essential. Saying Africans have the greatest human genetic diversity does not mean every African population has the same amount, or that all Africans belong to one ancient branch. Africa contains thousands of communities shaped by different languages, climates, migrations, and histories. Some experienced strong bottlenecks, some mixed with populations from Eurasia. Some expanded across huge regions, others remained relatively small and isolated. Two African populations can be more genetically differentiated from each other than a European and an East Asian population are from one another, depending on the groups and measures being compared. The first genetic tools that exposed this complexity were limited. Mitochondrial DNA follows a line passed from mothers to children. The Y chromosome follows a paternal line from fathers to sons. These markers helped researchers identify ancient maternal and paternal branches, but each represents only one narrow path through a person's family tree. A person has thousands of genealogical ancestors within a surprisingly short number of generations, yet mitochondrial DNA follows only one mother's mother's mother, while the Y chromosome follows only one father's father's father. Powerful conclusions required whole genomes. By the early 21st century, sequencing technology allowed researchers to compare millions of positions across human DNA. The result was a more detailed map of migration, population size, and mixture. In 2010, researchers published complete genomes from southern Africa, including a hunter-gatherer from the Kalahari and individuals from other regional communities. The genomes contained large numbers of variants that were absent from the reference data commonly used at the time. The message was immediate. Science had been trying to describe humanity while missing a major part of human variation. The people involved were often placed under the broad label Khoe-San, but that word can hide as much as it reveals. Khoe-San is commonly used as a collective term for diverse indigenous peoples of southern Africa, including communities historically associated with hunting, gathering, and pastoralism, and with languages containing click consonants. It does not describe one single tribe, one language family, or one genetically uniform population. San communities differ from one another. Khoe-speaking pastoralist histories are not identical to San histories. Northern and southern groups can have deep differences, and language does not always match genetic ancestry perfectly. This diversity became clearer in 2012, when genome-wide research on In 2017, scientists analyzed genomes from 7 ancient individuals in KwaZulu-Natal, South Africa. 3 Stone Age hunter-gatherers who lived around 2,000 years ago were genetically related to present-day southern sand populations. 4 later Iron Age farmers were closer to present-day Bantu language-speaking populations. The ancient genomes allowed researchers to examine southern African ancestry before some of the more recent mixing visible in living populations. Using those genomes and demographic models, the team estimated that one of the earliest population divergences among ancestors of living humans occurred somewhere between approximately 260,000 and 350,000 years ago. It overlapped with the period when fossils in Africa show populations developing combinations of features associated with early Homo sapiens. But this number must be handled carefully. It does not mean a modern sand community has existed unchanged for 300,000 years. It does not mean scientists found a pure bloodline stretching directly from the first humans. The estimate describes a population divergence in a model, not the birth date of a modern ethnic identity. The populations on both sides continue to change, mix, divide, and reconnect. Different models can produce different dates because ancient African population history was not a clean split followed by permanent isolation. Newer research has made the picture even less like a simple tree. Studies support models in which early African populations remained weakly connected over long periods. Gene flow may have continued after major divergences. A branch could separate, exchange ancestry later, and separate again. This creates deep genetic structure without requiring one population to have lived in complete isolation since the beginning of Homo sapiens. The human family tree inside Africa may look more like braided roots than separate straight lines. Even so, the southern African signal remains extraordinary. Many Khoe-San individuals preserve ancestry connected to lineages near the deepest known divergences among living humans. Their genomes contain variation that helps scientists reconstruct stages of human history poorly represented elsewhere. Recent ancient DNA work has strengthened the evidence for remarkable continuity in parts of southern Africa. In 2024, researchers published genomes from people buried at Oakhurst Rock Shelter in South Africa, creating a genetic timeline across much of the last 10,000 years. The results showed long-lasting continuity from early Holocene hunter-gatherers into later southern sand-related populations. That continuity stood out because many other regions experienced repeated, large-scale migrations and replacements. In southernmost Africa, the Oakhurst genomes suggested that a local genetic profile persisted for thousands of years before major outside ancestry became common. But once again, continuity did not mean isolation forever. The last 2,000 years brought enormous changes. Pastoralists carrying ancestry connected partly to eastern Africa moved south. Bantu language-speaking farmers carrying substantial Western African-related ancestry expanded into southern Africa. Later, European colonial settlement introduced another layer of ancestry, while bringing land seizure, forced labor, violence, and demographic collapse. Modern Khoe-San groups contain different proportions of these histories. Some identities were broken apart or reclassified by colonial systems. This is why describing one modern group as the oldest race is not only scientifically wrong, it can erase the very history the DNA reveals. Khoe-San peoples are not museum exhibits from humanity's childhood. They are modern communities whose ancestors traded, migrated, married outsiders, fought, adapted, and survived. They have histories of change just like everyone else. Deep ancestry is part of that story, but it is not the whole story. The maternal lineages add another famous piece of the mystery. Mitochondrial haplogroup L0 is among the deepest branches in the human mitochondrial family tree and occurs at high frequencies in some southern African populations. Because mitochondrial DNA is inherited through one maternal line, L0 has been used to explore ancient population structure and movement within Africa. Some studies have proposed specific southern African regions as possible homelands for this maternal lineage. But a mitochondrial branch cannot locate the birthplace of all humanity by itself. It represents one surviving line among many ancient lines, some of which disappeared. The woman from whom all living mitochondrial lineages ultimately descend was not the only woman alive in her time. She is simply the one whose unbroken maternal line survived to everyone living today. Other women contributed most of our nuclear DNA, even if their direct maternal chains ended. The Y chromosome tells a similarly dramatic but incomplete story. In 2013, researchers reported a rare Y chromosome lineage known as A0, with an estimated common ancestor far deeper than earlier Y chromosome estimates. The lineage was identified in men with ancestry connected to Western Central Africa, not specifically among the San. Its discovery showed why no single population can be called the sole keeper of ancient DNA. Different pieces of early human variation survive in different African communities. That is one of the greatest corrections to the original ranking idea. The deepest maternal line may be concentrated in one set of populations. A deeply branching paternal line may appear somewhere else. Whole genome diversity may be highest in another sample, depending on the populations, methods, and admixture being measured. There is no universal scoreboard on which every race can be placed from oldest to youngest. The word race creates another problem. Human genetic variation is real, and ancestry can often be estimated geographically. But traditional racial categories divide continuous, overlapping variation into a few social boxes. The boundaries change between countries and historical periods. A person classified as black in the United States may carry ancestry from several African regions, Europe, and the Americas. Two people placed in the same race may have very different ancestry. Two people placed in different races may be genetically closer than expected. Most human genetic variation exists among individuals, not between continental race labels. That does not mean population differences are imaginary. Some genetic variants occur at different frequencies because populations experienced different environments and histories. But those differences do not form separate human subspecies, and they cannot support a biological ranking of races. Human populations have always exchanged genes whenever geography and culture allowed contact. The same logic explains why more genetic diversity does not mean more intelligence, strength, or evolutionary advancement. A variant can be useful in one environment and harmful in another. Sickle cell variants can protect against severe malaria in certain genetic contexts, but can also cause devastating disease. Lactase persistence allows many adults to digest milk and became common in some pastoralist populations. Yet most adults throughout human history did not need it. High-altitude adaptations arose through different genetic routes in Ethiopia, Tibet, and the Andes. No single population collected every useful trait. Khoe-San populations also show signals of local adaptation, but sensational claims often go beyond the evidence. Researchers have investigated variants connected to metabolism, immune response, body composition, and life in dry environments. These patterns may reflect selection, drift, or both. They do not mean every person in a community has the same biological abilities. Population genetics describes changing probabilities, not fixed racial powers. The power of this diversity may appear in medicine. Genomic research has historically relied heavily on participants of European ancestry. As a result, tools for interpreting disease variants and calculating polygenic risk often work better for European-descended populations than for underrepresented groups. A variant that appears rare in a European database may be common and harmless in an African population. Without appropriate reference data, clinicians can misclassify it. African genomes are especially valuable because they contain enormous variation and shorter blocks of linked DNA produced by a longer population history and more recombination. These features can help researchers narrow the location of disease-related variants. Projects such as the African Genome Variation Project and H3 Africa were created to expand genomic research on the continent, train scientists, build local capacity, and improve health studies. The African Genome Variation Project analyzed dense genetic data from more than 1,000 people and whole genomes from hundreds across sub-Saharan Africa, finding novel variation and regionally distinct histories of admixture and selection. Yet, calling indigenous DNA valuable creates an ethical danger. For centuries, researchers collected human remains, blood, stories, and biological samples from colonized peoples without fair consent or shared benefit. San communities have faced exploitation by scientists who treated them as objects from the human past. Some studies used language that reduced living people to ancient specimens. Samples traveled overseas while communities received little control over how data were stored, interpreted, or commercialized. Modern research must do more than obtain a signature on a consent form. Communities should help decide the questions, control access, receive clear explanations, share benefits, and have the right to challenge harmful interpretations. The ancient Oakhurst Project reported consultation and ethical procedures involving community representatives and South African institutions. Such practices are essential because the deepest lesson of human genetics is shared ancestry, not permission to take from people whose history has already been taken in other ways. The question of archaic ancestry adds another layer. Non-African populations carry DNA inherited from Neanderthals, and some populations in Asia and Oceania carry substantial Denisovan-related ancestry. Africa also contains signals that may reflect ancient population structure or mixture with deeply diverged human groups. Researchers sometimes call these sources ghost populations because no confirmed reference genome exists. It is tempting to describe such signals as DNA from unknown species, but the models are debated. A pattern that looks like archaic interbreeding can sometimes be produced by long-lasting structure among early Homo sapiens populations. Without ancient genomes from the proposed source, scientists cannot always distinguish a separate archaic group from a deeply divided modern human population. Africa's climate makes DNA preservation difficult, leaving major gaps in the record. This uncertainty does not weaken the story, it makes it larger. Human origins were probably not one clean event. Early populations across Africa may have carried different combinations of traits and genetic variants. They interacted over long periods, and the population we call Homo sapiens emerged from that network. Southern Africa was one major part of the process, but Eastern, Central, Northern, and other regions also hold essential evidence. Fossils support this continental complexity. Jebel Irhoud in Morocco preserves early Homo sapiens remains more than 300,000 years old. Omo Kibish and Herto in Ethiopia preserve important early modern human fossils from Eastern Africa. Florisbad and other southern African sites add different combinations of features. No single fossil is simply the first modern human. The record shows populations gradually assembling the anatomy and behavior associated with our species. Genetics tells a matching story. The deepest branches do not point to one modern person as the unchanged original humans. They point to a long period of structure inside Africa before the major expansion outside the continent. Khoe-San related ancestry is crucial because it preserves one side of some of those deep splits. Other African populations preserve other branches, mixtures, and adaptations. Together they show that humanity's roots were broad, interconnected, and far more diverse than older textbooks suggested. So what happened when scientists supposedly ranked every race by DNA age? Nothing, because that study did not exist in the form described by the viral story. There was no credible 2019 project that sequenced every race, assigned each one an age, and placed a winner at the top. Large biobanks have analyzed hundreds of thousands of people, but sample size does not transform genetic diversity into a racial age chart. The real discovery came from decades of separate studies: mitochondrial research, Y chromosome analysis, genome-wide surveys, complete genomes, demographic modeling, and ancient DNA. These independent lines of evidence converged on several conclusions. Africa contains the greatest human genetic diversity. Diversity generally decreases with distance along ancient migration routes out of Africa. Several Khoe-San populations preserve ancestry associated with some of the deepest divergences among living humans. Southern Africa also experienced continuity, migration, and admixture, so no modern group is genetically frozen in the distant past. That truth is more powerful than the false ranking. Imagine the human family tree before the great migrations into Eurasia. It is not divided into white, black, Asian, or indigenous American races. Those categories do not yet exist. Instead, many African populations move through changing landscapes, sometimes isolated by deserts, forests, distance, or climate, then reconnected when conditions change. One branch contributes ancestry that survives strongly among Southern African hunter-gatherer descendants. Others contribute to populations elsewhere on the continent. Much later, a subset of this already diverse humanity expands beyond Africa and becomes the main ancestral source for most people outside it. Every person alive remains part of that earlier African diversity. Non-Africans did not stop being connected to Africa when their ancestors migrated. Their genomes carry a subset of variation from African source populations, later combined with Neanderthal or Denisovan ancestry and shaped by new environments. Many Africans also carry ancestry from population movements within the continent, and in some regions, from migrations back into Africa. The borders on a modern map cannot contain this history. The deepest branch is therefore not owned by one race. It is evidence of a shared past preserved unevenly among living people. A Khoe-San genome can help reveal a divergence that affected the ancestors of everyone. An ancient Southern African skeleton can change estimates for the entire human family tree. A rare Y chromosome in Central Africa can rewrite paternal history worldwide. A mitochondrial lineage concentrated in Southern Africa can illuminate ancient maternal movements across the continent. This is why the result should shock us, but not for the reason promised by the title. The surprise is not that one race defeated the others in a contest for age. The surprise is that the idea of separate races collapses when the full genetic record is examined. The greatest divisions in our species are ancient population branches, not permanent categories of worth. None remained pure. None evolved toward a higher level of humanity. The Khoe-San peoples did not shock science by being older than everyone else. They shocked science by revealing how much of the human story had been missing. Their genomes showed that early Homo sapiens history reaches deeper, contains more structure, and includes more variation than models built mainly from European and Asian data could capture. Ancient genomes showed continuity in Southern Africa that survived for thousands of years, even as later migrations and colonial violence transformed the region. The final lesson is not a ranking, it is a warning. When science studies only the populations with the most money, the largest biobanks, or the easiest access to laboratories, It can mistake a small part of humanity for the whole. For years, the standard human reference genome was treated like a universal map, even though much of global variation was missing from it. Every newly studied African population has the potential to reveal variants, histories, and medical information absent from older databases. And the deeper science looks, the less human history resembles a race with one winner. It resembles a web. The threads begin across Africa, divide, cross, disappear, and reconnect. Others cross Asia, enter Oceania, or reach the Americas. Some remain within Africa, carrying variation that other branches lost. All of them belong to one living species whose members are far more closely related than the language of race suggests. For decades, researchers searched genomes for the oldest surviving human line. What they found was not one pure person standing at the beginning of time. They found fragments of the beginning distributed across living populations, with some of the deepest and most diverse records preserved in southern Africa. The Khoe-San were not a window into a primitive world. They were a mirror showing modern humanity how incomplete its own reflection had been. Their DNA does not say they are more ancient humans. It says the rest of us forgot how ancient all humans are.